Hi, everybody. Good afternoon. All right, we're going to do the second part of our patterns of infection today, and we're going to talk about persistent infections, the ones that are never cleared. They last the lifetime of the host. So last time we talked about acute infections, which you'll remember are rapid, relatively rapid, and self-limiting. Persistent infections, if you remember one thing, is that they last the life of the host. You get infected, and they never go away until, of course, you die. And these are stable and characteristic for each virus. What I mean by that is every virus has its own kind of pattern of persistence, although we can put them in classes, as you'll see today. And in the course of a persistent infection, that doesn't change. It remains the same. And, of course, most persistent infections probably begin as an acute infection, which you may or may not see. They may be apparent or inapparent, and we'll give some examples of that today. And in fact, we'll see some examples of viruses that cause both acute and persistent infections. So here's our diagram of the general patterns of infection. Again, we're looking at virus production on the y-axis, time on the x-axis, and the virus production is in blue. Disease episodes are in red. And again, the acute infections, very defined periods of virus reproduction, disease, and clearance. These can be inapparent, of course. And then we have three different kinds of persistent infections. So you'll hear lots of different names for these. I like persistence. Some people use chronic infections. You'll see that as well. In my view, they're the same thing. And here we have three different kinds of persistent infections. Let's start with the one in the middle, persistent asymptomatic. This is an infection where it begins at a certain time. You have virus production throughout the life of the animal, but no disease symptoms, right? Obviously, there are signs because you could take blood from these animals and find virus in it, so that would be a sign of infection, but there are no symptoms. There's no overt disease, and the virus, nevertheless, is produced throughout the life of the host until the host dies, of course. And then on the fourth line, we have persistent pathogenic infections. These are simply lifelong infections where virus is produced throughout the lifetime, and there are only a few periods of disease. So the bottom one is HIV, where you have an initial infection, what looks like an acute disease. You have virus production, you have symptoms of that, and then the virus titers go down to a baseline. It's not zero. It is a baseline that can be measured for the lifetime of the individual. And again, this is untreated AIDS infection. 10, 15 years later, viral titers begin to increase as the system is completely destroyed. And then that's accompanied by disease and then death. And there are no other viruses that have a similar pattern as well. These are all persistent infections because that means they persist for the life of the host. Now, the second line has one called latent. I view this as a kind of persistent infection. It has a certain characteristic where there are episodes of virus production throughout the lifetime of the host, followed by periods where you can't detect any virus. And in fact, for some of these viruses, you can't even detect any viral proteins. The only thing you can find is the genome. And so we call that a latent state because the genome is not being expressed. So that's why you'll see them called latent, to emphasize the fact that the genome is present in a silent state, altering with productive states. But they're all persistent infections. And today we're going to examine latent infections. We're going to examine persistent asymptomatic and pathogenic infections as well. Now, a couple of general principles about persistent infections. These are the viruses we're going to talk about today. And maybe you could look at them and guess what they are. We have a Measles, a polyomavirus, Hepatitis B virus, a flavivirus, and herpes viruses. They all cause persistent infections. They occur when the primary infection is not cleared by the immune response. So you have an initial primary infection, which you could view as an acute infection, and the infection is never cleared. So the genomes remain for the life of the host. And viruses, proteins, genomes continue to be produced in some cases, but we'll see in other cases only viral genomes are present and they're not expressed, no proteins are present. So the viral genomes may remain after proteins are not detected. This varies according to the virus. Some viruses, all of these, the genome is present clearly. For some, there are episodes of virus production, and for others, there's continuous virus production. But they all seem to start with an acute infection that's not cleared, and that should suggest to you that there's some kind of antagonism going on, and that's one of the mechanisms for establishment of a persistent infection. There's no single mechanism. There are lots of different ways that this is accomplished. We don't understand them by any means completely. I'm going to tell you a little bit of what we know today and a lot of hypothesis, but there's still a lot to be learned on the specific mechanisms. And it's safe to say that even within a virus family, there are going to be different mechanisms for persistent. And one of the key properties in initiating a persistent infection, we don't have cytopathic effects, right? And that, remember, that's important for inducing inflammation. If a virus doesn't kill cells and release proteins that can be recognized by dendritic cells, so you get a good adaptive response. If you don't get cell killing, you're going to get a poor host defense, and that leads to a persistent infection. Virus immune modulation is also another component of having a persistent infection. You'll see that, and we've already talked about this, the various ways that viruses can antagonize specific parts of the immune response, and this is clearly plays a big role in establishing persistence, and we can show in animal models if we take out these immune modulation mechanisms, animals are not persistently infected. So here are some persistent human infections that we know of. The viruses on the left. The place in our body where these viruses persist are shown in the middle, and the consequences are shown on the right. So you can see we have a whole range of viruses. Adenoviruses, we have a lot of herpes viruses. Here we have hepatitis B and C virus, HIV, of course, polyomaviruses, papillomaviruses, measles, and another herpes virus at the end. You can see some of these have no known consequences, and others have very serious consequences as well. And the cells that can be infected are quite varied, from cells of the immune system to epithelial cells, cells of various organs, including the CNS. Today we're going to talk about the ones that have a red asterisk next to them to give you an idea of the different kinds of persistent and the different mechanisms that are involved. We're going to talk about HIV on its own towards the end of this course. It's the only virus that gets a lecture on its own. You know, I don't like to teach by virus because that's not how you learn principles of virology. You just get taught a collection of facts. But HIV is so important that we need to talk about it on its own. And one day, it may be preventable by vaccination, and it may become part of all the other lectures, and we'll have a different virus to take its place. So let's go through some of these. Now, an important part of persistence, as I mentioned, is viral antagonism of immune responses. And a major one is at the level of cytotoxic T lymphocyte response. Remember, these are what are important, crucial for clearing many virus infections. It's not the antibody response, that comes up too late, but it's the CTL response. And so if a virus can antagonize this response, it has a good chance of setting up a persistent infection. So CTLs, remember, recognize virus-infected cells. These infected cells have MHC molecules on their surface, which are displaying viral peptides that have been produced from viral proteins that are being made in the infected cell. The viral peptides are these little orange blocks. They're displayed in the MHC molecule. And the CTL, the cytotoxic T lymphocyte, will recognize that peptide by virtue of the T cell receptor. And there are other T cell surface molecules that are involved in this. And if it's a foreign peptide, the CTL will kill this cell. And so we have talked very briefly before about this pathway, but I just want to go over it again and point out all the different ways that we have learned that viruses antagonize MHC1. MHC1 is the endogenous pathway for antigen presentation. All that means is that the virus infects a cell, proteins are being made in the cell. That's the endogenous part. And then they get chopped up and put on the surface. That's in contrast to MHC2, which is the exogenous pathway. It pulls in peptides from outside the cell that are being released from, say, infected cells. And monocytes, dendritic cells are very good at that MHC2 system. So here we have a virus-infected cell. Here's a viral protein being made. These proteins get chopped up by the proteasome. They're targeted to go to the proteasome because they get ubiquitinated. They get covalently bound to a small protein called ubiquitin. And then peptides are produced. These peptides are imported into the endoplasmic reticulum by a transporter protein. They are then loaded onto MHC molecules. These MHC molecules, of course, are produced by translation of mRNAs on the endoplasmic reticulum. So they originate by transcription of mRNAs in the nucleus. The mRNAs are exported, they're translated, they're loaded with peptide in the ER, and then they're transported up to the cell surface. So the MHC1 molecule on the cell surface is then explored by the T cell receptor on the CTL, and if it's foreign, of course, the cell will be killed by mechanisms that we talked about. Now look at all these yellow boxes. These are places where viral proteins have been shown to block this MHC1 pathway. It's really amazing how many different mechanisms are at play, starting from inhibition of MHC transcription in the nucleus. There are adenovirus and HIV proteins that specifically inhibit that process. There are viral proteins, and here's an HIV protein that inhibits export of MHC mRNA out of the nucleus. There are viral proteins that block the transporter of the peptides. There are some viral proteins that pull the MHC out of the ER and degrade it in the cytoplasm by the proteasome. Instead of allowing the MHC to go to the surface, these proteins cause it to be directed out. We have an Epstein-Barr virus protein that it inhibits processing by the proteasome. We have other viral proteins that inhibit transport of MHC1 to the cell surface. And finally, some viral proteins that take the MHC1 that's on the surface and bring it back into the cell. They downregulate surface MHC by either pulling it into the cytoplasm or bringing it to a lysosome where it's degraded. Of course, unless MHC is on the surface, it can't do any good in the T cell recognition pathway. So all of these mechanisms will allow a virus-infected cell to escape killing, allows the infection to proceed, and that can lead to persistence. So we can take away these genes from these viruses and show they have no role in cell culture whatsoever. They are specific for infection in an animal to antagonize the cellular immune response. Messing with the MHC1 pathway is very important, but there are other ways of evasion of MHC1 pathways, and that's by what we call CTL escape mutants. So remember, in the cell, in the infected cell, the viral proteins are chopped up and loaded onto MHC molecules. And so here in the middle of this diagram on the left, we're showing a peptide with the little green amino acid. So, not many amino acids are interacting with MHC. So MHC is the gray molecule, and the green is the viral-derived peptides with individual amino acids binding nicely. But these viral sequences can change. Viruses undergo mutation, of course. And single amino acid changes are enough to block binding of the peptide to the MHC molecule. So as viruses replicate, variants arise that have changes in these T cell epitopes, and those changes will block binding of the peptide to the MHC, so it can't be presented on the surface of the cell. So that's just another way of evading MHC1 mediated killing. This is by viral variation in the epitopes that are loaded. So one mechanism is that the changes in the viral sequence prevent binding to MHC. These changes can also affect proteasomal processing. They can make the viral protein precursors resistant to proteasomal processing, so they can never get chopped up and loaded into MHC. So these mechanisms have been very well studied for herpes simplex virus and hepatitis C virus, both of which cause persistent infection. So again, another very clever way of antagonizing this killing by preventing loading of the protein into the MHC1 molecule. Yet another interesting mechanism. Here we've been talking about a CTL killing an infected cell, a virus-infected cell. Sometimes the infected cell can produce products that cause the CTL to die. So instead of the CTL killing the infected cell kills the CTL, turning the tide, if you will. So here on the left, CTL killing an infected cell. Now these CTLs on their surfaces have a variety of receptors, which are shown on the right here. One of them is a receptor for Fas ligand. And binding to all of these receptors by their ligands induces cell death via apoptosis. Now, Fas ligand binding to its receptor on the surface of the T cell will cause the T cell to die. This is a normal part of cellular responses to limit immunopathology. So when you have a virus infection, CTLs are produced and recruited to the site of infection. At some point, you don't need them anymore. Remember, we talked about how CTLs cause immunopathology on their own, so you don't want them hanging around forever. If the viral infection is under control, you want to get rid of them. So what you do is you make Fas ligand to kill off the CTLs. So this is a normal immunomodulatory mechanism. Some viruses have evolved to produce a lot of Fas ligand in the infected cell, which goes out, binds the Fas ligand receptor on the CTL and kills the CTL before the CTL can kill the infected cell. And HIV and cytomegalovirus can both induce the production of Fas ligand on the cell surface to do this. All right, so that's another example of viral defense producing Fas ligand. Now remember, Fas ligand and its receptor is going to come into play in the very next slide, in fact. The final point I want to make about persistent infections is that they also arise because there are certain areas of our body that don't have such great immune defenses. Because if they did, it would not be good for us. These include the central nervous system. Right? Your brain is in this hard shell. And if there's too much inflammation in your brain in response to infection, the brain's going to swell and run out of room to expand, and this is not good. This is why brain infections are bad in the first place. But we don't have a huge immune response in the brain because it would be bad to do so. It's limited to cytokine production, trying to clear infections that way. So the CNS, the brain in particular, is immune modulated. There's not a huge immune response. The vitreous humor of the eye. So here's the eye on the upper. The vitreous humor is the liquid in the eye proper. Same problem you have with the brain. If you had this huge inflammatory response, your eyeballs would swell and would be damaging. So the vitreous humor of the eye has a reduced immune response. We call these immune privileged areas. There are a couple of others. Areas of lymphoid drainage are also devoid of initiators and effectors of the immune response. The eye in particular has high Fas ligand in it. What does that mean? So if any CTL came in to try and get rid of a virus-infected cell, the CTL would be killed before it could do so and cause cell damage. Which is not to say that things don't happen in the eye caused by virus infections, but they are much, much lower in terms of immune responses. And as a consequence of all this, we often get persistent infections of the CNS, of the eye, and of other places where we have this reduced immune surveillance. A couple of years ago, I was talking to my class about this, how the eye is an immunoprivileged site. And then the next month, this article came out in the New England Journal. It was in the midst of the Ebola outbreak in West Africa. And what better example to tell you what I was talking about was this paper finding persistence of Ebola virus in ocular fluid. A number of individuals got infected working in West Africa. They came back to the US. In fact, it was an emergency room physician from Colombia who came back from his work in West Africa, and he developed a fever here, contracted Ebola while he was here. So he was hospitalized and he recovered. But then months later, he started having eye issues. And there were several other cases as well where people had eye issues, and it turned out the virus was replicating very, very slowly in the eye because the eye is an immunoprivileged site. It was never eliminated from the eye. Apparently virus got in, it didn't leave, okay? And you don't look in the eye for virus. When you say you're free of virus, where do you think you look? In the blood. You take some blood, you do a PCR, no signal, okay, you can go home. The virus is still there because the eye is an immunoprivileged site. So we're learning that this virus, which we always classified as an acute infection, as causing acute infection, can also cause persistence, not only in the eye, turns out, also in the testes, another immunoprivileged site, tightly contained. You don't want to have an excessive inflammatory response there. They're finding Ebola virus in testes long term. Now, the significance of that isn't known because people don't typically look for infectious virus. They look for pieces of nucleic acid by PCR. So we don't know if it's actually infectious or not. It's very hard to do an infectivity assay with Ebola. You need to use a BSL-4 facility. And recently, the same has been found for Zika virus. It seems to persist in the testes of men for months after they are infected. Again, how long it's infectious virus, we don't know. But the point here is that what we think are acute viruses can also cause long-term infections. And, you have to look. You're not going to find anything unless you look. We're lucky that these individuals had eye symptoms. They were having eye conjunctivitis, essentially. Their eyesight was funny. And when the doc looked in the eye, he or she could tell that there were problems caused by infection. A nice example of what I'm talking about. Many viruses, as we talked about some time ago, can infect immune cells. This is another way that persistence can happen because when you infect immune cells, obviously you disrupt immune responses. And so many viruses infect these leukocytes that we've talked about in the last few lectures. Measles virus, for example, infecting antigen-presenting cells. And as we'll see, HIV infects so many immune cells. That's why at the end of 10 or 15 years, you get all sorts of opportunistic infection. You have no way of responding to infection any longer. So direct infection of immune cells is another way to establish persistent infections. All right, so that's an overview of all the different ways that viruses can hang around. Our first question is, which of the following are features of persistent infections? They last the lifetime of the host. Viral immune modulation is involved. Immune cells may be infected. They may occur in areas of reduced immune surveillance. All of the above. There you go. Answer is E. Everybody got it. So I think you're now the best class ever in terms of getting 100%. So let's talk about some specific examples of persistent infections. One of them is measles virus. And you remember last time we talked about measles virus as the cause of an acute infection. Measles, of course. Paramyxovirus, one of the most contagious human viruses we know of, with an R0 of 15. 115,000 deaths in 2014, all of them preventable, not everyone vaccinated. That's why we have so many deaths. Lifelong immunity after infection. Classic acute infection, as we talked about last time. However, there is a long-term sequelae of infection called SSPE, which I mentioned last time. Subacute sclerosing panencephalitis. This is a progressive degenerative encephalitis. So these are kids that have measles, and 1 in a million of them will go on 6 or 8 years later to develop SSPE. So these kids will seemingly be fine, and all of a sudden they have cognitive and motor degeneration. They get worse and worse, so it's a progressive neurological disease, and it's uniformly fatal. There's no way we can treat this. All of these kids die. And they are all preventable by measles vaccination. So 1 in a million is not a lot. Would you like that to be your kid? No, I wouldn't, because this is a nasty disease. So I think people who refuse to immunize their kids against this virus should really understand how bad this can be. So what is happening? We really don't understand what's going on. All we know is that you can find nucleoprotein particles in the brain. Remember, the nucleoprotein is the RNA complexed with proteins. It's a negative-stranded RNA, so it's complex as a nucleocapsid in the particle. We can find these in the brain. There's no infectious virus, but these nucleoproteins with the viral genome can go from cell to cell within the brain by crossing synapses, which suggests that they can bud out of cells and get into new ones. So again, there's no infectious virus, but there's clearly viral genomes there, and that goes along with the disease. We simply do not understand what's happening. There's no good animal model for understanding this, and it's so rare in people that we can't really understand if it's a genetic problem in these individuals or not. Nevertheless, it's an example of how there can be a very common acute infection leading to persistence. Again, these are 6 to 8 year incubations leading to virus until the children die. The next group of viruses are the polyomaviruses. This is a family of viruses we've talked about a lot in this course. Remember SV40 we used as a model for understanding DNA replication and transcription. Remember the T antigen? T antigen is going to come back. Don't forget it. Right? It's going to come back in next lecture, actually, in a really interesting way. This is a monkey virus, simian virus 40. But there are all kinds of related viruses in this family, polyomaviruses. You can see there are a bunch of them here. This is divided into 3 genera, and one of them is called orthopolyomavirus. You can see there's a bat polyoma in there in SV40. These ones with the red asterisks are human polyomaviruses. BK polyomavirus. This is JC. It's actually a man's initials. And then a few others. Merkel cell polyomavirus is one that causes a rare skin tumor in humans. Then we have some bird polyomas. And then we have a series isolated from Wookiees. Can you imagine? They actually got viruses from Wookiees. Nobody ever laughs at that from year to year. You're like, what is he talking about? They're called Wookiee polyomaviruses, see? The name is coming from the fact that one of them is called WU from Washington University. That's where it was isolated from a patient. Another one from the Karolinska Institute, KI, Wookiee polyomaviruses. These are in all of you. Every one of you, I guarantee, has some kind of polyomavirus in you. They infect you very early on in life. Serological studies have been done in different populations, and you see very high seropositivity, depending on which virus you're looking for out of all those human viruses, up to 99%. So that's a pretty good shot that all of you are seropositive. And these viruses replicate in a variety of organs in us, kidney, intestine, respiratory tract. You pick them up probably early in life from your parents, your family. They replicate in you, and for the most part, there's no pathology. The one thing I like to point out is that from time to time, you can have a burst of replication in your kidney. It's not clear why you have this. And then you excrete up to 100,000 particles per milliliter of urine. So just think of that next time you go to a public restroom. Toilets and urinals generate aerosols. So you're inhaling someone else's polyomavirus. It's another thing to put on your bucket list of things not to do. Don't go to public restrooms. Well, you can go, just be careful. You know, in fact, it's probably not a problem to pick up someone else's polyomavirus because they're benign for the most part. But I just want you to know that the air in the restroom is full of polyomaviruses, if you didn't know that before. We don't know how these persist. People are working on it, but I can't give you any nice scenarios like I'm about to do for other viruses. We do know one case where they cause problems, and that is people who are immunosuppressed. So there is, as you know, there's a disease called multiple sclerosis, and one of the treatments for multiple sclerosis is a monoclonal antibody that's immunosuppressive. It's directed against an antigen, I think, on B cells. And since all of us have these viruses in us, as soon as we get immunosuppressed, they begin to replicate out of control in certain tissues, including the brain. That's where PML results. And so in people being treated for multiple sclerosis, a number of them get this disease, a progressive fatal disease, again, suggesting that persistence is maintained somehow by immune cells keeping the virus in check. If you want to learn more about these cool viruses, I did a TWIV with a doctor out in Colorado, and he's worked on these all his life, and he had a lot of cool things to say. We called the episode Wookiee viruses, of course. Okay, our next virus, Hepatitis B virus, which again, you all remember because most of you didn't get the answer right on the exam, why it has DNA as opposed to RNA in its capsid. Okay, but we talked a lot about Hep B because it's a DNA virus with a reverse transcriptase in its reproductive cycle. The answer, by the way, is that this thing packages RNA in the cell and it reverse transcribes it before it leaves. Very simple. So there's the DNA genome. Remember, it's gapped and it's got a piece of RNA and a protein on it. Well, this virus is transmitted among people in a number of ways. Exposure to blood, and that can happen during childbirth, a lot of blood there. You get a blood transfusion that's contaminated, sex, drug use, tattooing. Be careful when you get tattoos. If you want a tattoo, go to a doctor or a nurse who's going to sterilize their instruments, because this is a big way that HBV is transmitted. as well as some other viruses, as you'll see, and also nosocomial in hospitals. Blood supply should be clean of Hep B because we tested, but before we knew about it, we were transmitting these viruses via the blood supply. The main target's the hepatocyte, so replicating mainly in liver cells. Most people who are infected, 95%, can clear an acute infection that results. I'll show you what that looks like in a minute. However, newborns have a lot of problem resolving infection, which you might guess because they don't have much of an immune system, so they have typically more problems. And so you can get infected at childbirth, and so that's an issue. So here are two typical profiles of Hep B infection. A lot of people worldwide have chronic Hep B infection, about 350 million people. Chronic means persistent. They have it for their lifetimes as far as we can tell. And the big issue here is a fraction of them will develop liver cancer as a result of the virus replicating in liver cells and the immune destruction that results. So on the left is a typical course of acute hepatitis. So here we have exposure at time 0, and then weeks are on the x-axis. And we're looking at a variety of measurements on the y-axis. One of them is one of the proteins of the virus, hepatitis B surface antigen, is in red. You can see it peaks maybe at 9 weeks or so, begins to decline. Viral DNA is in green. You can see it have a defined peak and a clearance. By a year, it's gone. So it's a long-term infection, but it is a defined infection. You make antibodies against various viral proteins, and symptoms are shown here in this blue box, so a defined time of symptoms. And those are accompanied by release of enzymes from the liver, like alanine transaminase, ALT. This is an enzyme which should not be in your blood. If it's there, it's because your liver cells are being damaged. So you can see a defined peak of ALT production. This is an acute infection where it's resolved. The immune system can take care of it. Virus is gone after a year, and you're fine. However, in a number of situations, the virus goes on to chronic hepatitis B. You have, again, an infection, but the viral protein, Hep B surface antigen, you can see continues for a long time, for the life of the individual. Viral DNA is present for the life of the individual. ALT is present for the life of the individual. All right, and eventually these individuals are going to have severe liver damage, which is a problem in itself. You compromise liver function, and of course, liver cancer. Now, the interesting part here is that the virus is not killing hepatocytes. It's not cytopathic for hepatocytes. So what's causing liver damage? Cytotoxic T lymphocytes are killing the infected liver cells. So they're causing the liver damage that releases the ALT and other liver enzymes into the blood. What we think is that in people who never resolve infection, who go on to be chronically infected for their entire life, there's so much T-cell, CTL activity, they enter a state that we call T-cell exhaustion, where they can no longer respond to foreign antigens, so they lose their functions and they can't clear the infection. So these individuals, so the points here are that the CTLs are causing the damage, the hepatitis that leads to liver dysfunction, and then their exhaustion leads to dysfunction, so the infection can't be cleared. We're not really 100% sure about this. This is a hypothesis. And during chronic infection, you get liver fibrosis, destruction of cells and production of new cells, eventually cirrhosis and liver failure. Now, hepatocellular carcinoma, liver cancer, will develop after about 20 to 30 years of chronic infection. And that's a fatal disease, so you don't want to have liver cancer. And this is often asymptomatic. If you go back to this previous slide of Part B, chronic hepatitis, you see the symptoms are in a defined period, and then after that, there are no symptoms. Of course, you have signs of infection. You have elevated ALT, viral antigens and DNA in your blood, but you feel okay, so you don't have any symptoms. And then eventually, years and years of this, 20 to 30 years, you have liver failure and death. So there are some antiviral drugs available to treat these infections. They're not very good. We need better ones. Some vaccines are in development as well. But you know, the best way to take care of this is to prevent infection in the first place. All right, another virus that's causing liver infection is an RNA virus, so hepatitis B. I call it a DNA virus because what's in the particle is DNA. It has an RNA phase and a reverse transcriptase phase, but this is an RNA virus. It's a flavivirus, positive strand RNA genome within a capsid that in turn is within a membranous shell, and then within that are the glycoproteins that allow the virus to attach to cells. So many flaviviruses are transmitted by arthropods, like Zika virus and West Nile virus, mosquito vectors. This one is transmitted from person to person by, again, contaminated blood. Same risk factors as hepatitis B virus, sex, drug use, tattooing, birth as well. And also, unfortunately, medical intervention, nosocomial infections as well. Same for Hep B. And so this is another hepatitis virus. So we discovered them consecutively, A, B, C, D, E, and then G. For some reason, we skipped F. And we discover them, and then we start checking blood for them. And then we see another one pop up that was masked, and we take that out of the blood. We check blood for that. So one by one, we've gotten rid of all these viruses from the blood supply, which was the main way of transmitting them. Now these other routes are quite frequent. 2.2% of the human population, 185 million people, are currently infected. So here's a global serological survey of people for antibodies to the virus. And the darker colors are higher, over 3.5% of the population seropositive. Again, you take blood from people, you look for antibodies to the virus, and you can see the northern parts of Africa, Middle East, Asia are all very highly seropositive. Other parts of the world, moderate to low seropositivity. Unfortunately, some of these very high regions, like Egypt, for example, is high because they were trying to eliminate other infectious agents, parasites specifically, during the 70s, and they used contaminated needles. They didn't properly autoclave them. They weren't plastic needles. They were glass, disposable needles. They were contaminated, and they passed hepatitis C from person to person. So they're trying to eliminate one infection, and by using contaminated needles, they spread Hep C to like 60% of the population. And so that's why Egypt, and I think many of these northern African countries also have similar problems. We're going to see how that kind of practice probably led to the emergence of HIV in the 20th century. So here's the course of hepatitis C. You get infected, you have an acute infection initially, where you have hepatitis, elevated liver enzymes in the blood, you get jaundiced, the whites of your eyes are yellow, classic hepatitis. 3 to 6 months, 10 to 40% of people will recover, resolve the infection, that's it, the virus is gone. However, 60 to 90% of individuals have a persistent infection where their liver is chronically infected. They start to have other problems. There's some reports of depression caused by infection, possibly by cytokines or other viral proteins entering the CNS, and other symptoms of infection like vasculitis and porphyria cutanea. And then over the next 30 years, 70 to 98% of those individuals will continue with an asymptomatic persistent infection, but 2 to 30% enter end-stage liver disease, which includes cirrhosis, serious cirrhosis, hepatocellular carcinoma, the development of fluid in the abdomen, ascites, jaundice, infection of the brain, encephalopathy, and esophageal varices. So this is a serious disease as well if it continues for many, many years. Now at the bottom is the course of infection in the two categories, acute versus persistent. The acute is in red, so we have introduction of virus here on the x-axis, which is time. On the y-axis, we're looking on the left at viral RNA in the serum, and on the right at a liver enzyme in the serum. So you can see in the acute infection, the red is viral RNA, peaks in about a month, then rapidly declines. The ALT also peaking and declining. And again, those are these 10 to 40% who recover and resolve. So in them, it's an acute infection. In others, 60 to 90%, the viral RNA persists at high levels for years after infection, and the ALT stays at high levels as well. We don't really understand completely why hepatitis C virus persists. People who clear it So that fraction of people who clear the virus after the acute infection, people have done genome-wide association studies, GWAS on them. They sequence their genome and say what's different about them from the rest of the population. It's not an easy exercise because there are a lot of bases in our genome, but you can focus on immune-related genes, and when you do that, you find interferon lambda 3 alleles seem to be, specific alleles seem to be correlated with protection. And so interferon is important for resolving infection. But the virus encodes multiple evasion mechanisms. It can evade at many levels of the interferon response in amazing ways by cleaving proteins with viral proteases and so forth. So it's a very difficult virus to clear. Those people who clear it early, they can, apparently their innate system gets hold of infection early on, but the ones who have persistent infections, they probably have a suboptimal interferon system, which remains to be fully characterized. We used to treat these individuals with interferon, okay? Years and years of interferon treatment. What do you think about that? Would you be happy doing that? No, you'd feel pretty crappy. And a lot of them would stop taking their interferon and get sicker and sicker. But now we have antiviral drugs. In fact, we have many of them that we can use in combination. So this infection could be eradicated. It only infects people, and the combination of drugs, 3 different drugs, avoids resistance. You can actually clear infected people totally if you give them the drug. Unfortunately, it's really expensive, so that's part of the problem as well. But maybe in the next 10 or 20 years, this could be eliminated. Of course, if people continue to engage in risky business and transmit virus, there's not going to ever be clearance, but theoretically it's possible. So here are the big 3 viruses that we've talked about. 2 of them, Hep B, Hep C, and HIV. This is the age-adjusted mortality rate from 1999 to 2007. You can see HIV is declining because of a variety of practices, informing people of their risk, triple therapy, and so forth. Hepatitis B is declining, but Hep C is rising still. And so this is before the onset of these new antivirals. I don't have any more recent data, but I would predict that it would level out and eventually decline. The next question is, which are shared features of persistent infections with polyomavirus, HBV, and HCV? The genomes are present but not expressed. Liver damage, kidney damage, virions are produced, all of the above. The answer here is D. Virions are produced in all of them. Okay, now I want to spend the rest of the time talking about a subset of persistent infections that are called latent infections, because they have a phase where there's no virus produced, that the genomes are still present. So here are some of the common features of these. And these are all going to be herpes viruses now for the remainder of today. Viral gene products that promote replication are not found, or they're very low. So the proteins we've talked about before, the early proteins that are needed for DNA replication, they're not going to be present. Cells that contain the latent viral genome. So latent means it's not expressed or poorly expressed. All right, you may have a few genes expressed, but certainly not the whole complement of genes, so we call that latent. And these are not well recognized by the immune system, so they can last your lifetime. But the viral genome is intact. The whole genome is there, and so that it can be activated so you can make new virus particles and spread to a new host. There would be no point in a virus establishing a persistent infection if it could not spread to another host, because when that host died, that would be the end of it, and that virus would eventually be gone. The genome state differs among these herpes viruses in interesting ways. In some cases, it's a non-replicating DNA in a cell that doesn't divide. So herpes simplex virus, Varicella-zoster virus, becomes latent in neurons, as we'll see, and it doesn't replicate. It doesn't have to replicate because neurons don't divide. So one genome in a neuron is plenty to keep the virus around. For some viruses, Epstein-Barr, cytomegalovirus, human papillomaviruses, which we're not talking about today, hepatitis B, Kaposi's sarcoma herpes virus. It's an autonomous self-replicating DNA in a dividing cell. So the cells are dividing in which these viruses are latent, so the genome has to divide along with it. And in one case, which we'll talk about briefly, human herpes virus 6, the viral genome integrates into the host DNA. And so that solves the problem of it replicating. It replicates whenever the cell divides. So first, herpes simplex viruses. We did talk a lot about these in terms of DNA replication and transcription, the coordination of gene expression. These are enveloped DNA viruses with double-stranded DNA, very large genomes that encode a lot of proteins. People in the US, over 80% of them are seropositive for these viruses, and that includes you, so 80% of you are seropositive, and you have genomes in your peripheral nervous system. You have viral DNA, every one of you. Now, you know you all have endogenous retroviral genomes in you, but now you know you have polyomaviruses, and you have herpes viruses in your peripheral nervous system. Millions of people have no symptoms throughout their lifetimes, despite being infected early on and having viral DNA in their peripheral system. They don't get disease, but 40 million people in the US experience recurrent herpes disease. That is, the production of lesions in the genitals or in the mouth, which are representative of viruses replicating. Some people have every 2 or 3 weeks regular production of lesions, which then heal. Of course, these lesions are producing viruses so they can infect other people. There are 2 different serotypes of herpes simplex viruses, 1 and 2. One is thought to be present mainly in the oral cavity, 2 genital cavity, but they can mix up as well. This is an example of a well-adapted pathogen. Not a lot of serious disease, infects a lot of people, and effectively transmits from one person to another. Here's how the infection works. So typically, you get infected initially either in utero. If the mother is infected, she may have genital lesions, and viruses being produced can infect the baby in utero, or at birth, the baby will be infected. In fact, 80% of babies are born infected with herpes simplex viruses. After a brief incubation period, the virus is replicating in initially mucosal epithelial cells. A lot of these infections are in a parent, but sometimes the babies can have various symptoms, fever, sore throat, lesions of various sorts, swollen lymph nodes, etc. The virus infects the epithelial cells, typical in the oral cavity, and the virus replicates in them, can spread laterally in the epithelium. These viruses can fuse from cell to cell. The viruses are also released into the subepithelial space, where they can find a variety of nerve endings. Here we're showing sensory nerve endings, which are just below the skin, of course. The virus can enter the nerve ending. It can be transported up to the cell body, where the nucleus is, of course, and there it can become latent. We'll talk in a minute exactly what latency means. So you have an initial acute infection, the virus goes into the cell body of the neuron, and then it establishes latency. These viruses can also infect neurons of the sympathetic nervous system, which are shown here. In both cases, the virus enters the nerve endings, makes its way to the cell body where there's the nucleus, and becomes latent. And there it's hidden from immune surveillance. In fact, it's not making any proteins. Herpes simplex viruses are not making any proteins, so the immune system doesn't even know that it's present. So that's shown on this slide, what happens after these initial infection events. So here we have our mucosal epithelium where the infection began. The virus goes down the neuron, the axon of the neuron, enters the neural nucleus, and is present as episomal viral DNA. All right, it's a piece of DNA apart from the chromosome. It doesn't integrate. It's silenced. It's coated with nucleosomes. Remember, it's wrapped around nucleosomes tightly so no mRNAs are made. It's silenced. There are multiple copies in the nucleus, and because neurons don't divide, the viral DNA doesn't have to replicate. It just stays there forever. You're stuck with it forever. Unlike love, herpes is forever. Right? It lasts your entire life. You cannot get rid of it with drugs or vaccines because the DNA is in there, and unless you want to get rid of your peripheral neurons, you can't get rid of it. This genome, by the way, is almost silent, doesn't make any mRNAs. It makes a small RNA called the lat RNA that's processed to microRNAs. We think that's in part required for latency. There's also a host contribution to keeping the genome silent, as you'll see in a moment. Upon stimuli, which we'll get into in a moment, a small number of neurons in the ganglia. Now, the ganglia, of course, is a collection of neuronal cell bodies, right? You have ganglia throughout your body where there are a bunch of cell bodies together, and so the herpes viruses have gone into these ganglia. They've infected multiple neurons within the ganglia, and they reactivate upon a stress. The DNA replicates, proteins are made, virus particles are assembled and released at the end of the nerve. They travel down the nerve back to the tissue, and the viruses appear in mucosal tissue. And sometimes you get a cold sore or fever sore, and that's the sign that herpes has come out of your ganglia and is replicating and making that sore. And of course, there's virus in the sore, and that's how you transmit it to other people. But not everyone has a fever sore, yet they're still shedding herpes viruses. Okay, so that's silent transmission to other people. And this virus is transmitted by intimate contact at any age of life. Now, you may say, why doesn't the immune response kick in? It's too slow. Virus encodes lots of antagonists, so there's not enough time before the virus is shed. Eventually, the infection is controlled. The fever blister goes away, and there's no more virus. That's thanks to our immune system. But it's delayed just enough so that the virus is shed and can be spread to another person, which is, of course, the modus operandi of the virus, to get to another host. And as I said, some people never reactivate, and some reactivate every 2 to 3 weeks. So if you initially are infected as a baby in the oral mucosa, the virus will enter the nerves that innervate that area. In your face, there are 3 principal nerves innervating your face below and above the jaw and your forehead and nose. So let's say you get an infection in the mouth. The virus will enter these nerve endings, and they go up to the trigeminal ganglion, it's called, because it serves 3 nerve branches. And that's where herpes is going to be latent in you. So if you want, you can have this removed and get rid of herpes. Some people have a disease called trigeminal neuralgia, where their face really, really hurts because of this nerve, and they actually have the nerve and the ganglia excised, and that can get rid of the herpes because they're lying in there in the nuclei. And then when reactivation occurs, the virus comes back out. It can go to the lower lip, it can go to the upper lip. Some people even get blisters on their nose as well. So that is reactivation. Now what happens during reactivation? So we know that stresses, sunburn, physical or emotional stress, nerve damage, hormonal imbalance, certain steroids will cause reactivation of herpes, replication, the production of virus, and the formation of a blister, like preparing for an exam. That kind of stress in many people causes the production of fever blisters. And what happens is this stress somehow produces the proteins needed to activate the transcriptional program. So remember, this viral genome goes through a series of transcriptional steps. VP16 initially comes in with the virus particle, activates immediate early transcription, which activates early and then late transcription. You have to go through an ordered series of events, right? But think of this. Now, in this neuron, you have the whole viral genome there. Where are they going to get VP16 from? That's only in the virus particle. Remember, it's in this area around the capsid. So this was a big problem for many years. I think it's been sorted out. Here's the answer, which has come from studying animal models and neurons in culture. What we think happens, here we have our neuron with a latent herpes DNA in it. But this genome is silent. Why is it silent? Because it's wrapped around chromatin, and the chromatin is methylated on very specific positions. Remember, methylation of histones causes chromatin to be tightly wrapped around the DNA so it can't be transcribed. And so we know that herpes DNA in these ganglia is extensively methylated on histone 3, lysine 9, histone 3, lysine 27, methylation on different positions. So all the promoters are shut off upon application of a stress, whatever it's going to be, sunlight, a test. You have activation of protein kinases in the central nervous system. These phosphorylate these lysines that are methylated. So the phosphate is shown in this second panel as a blue circle. So the methylation is the red, which shuts off transcription. Turns out that the phosphorylation can turn on all of the herpes genes. It can reverse the silencing caused by methylation of histones. Just putting phosphate on. So you don't have to actually remove the methylation, which is usually what you do to turn on genes. You remove the methylation. But here, apparently, phosphorylation, and with this phosphorylation is by a kinase called JNK, which is upregulated upon stress. Perfect, right? So the genome is all expressed at once. All of the genes are produced. They're all transcribed, immediate, early, and late. And guess what gets made? What gets made? VP16. that gets made, which you need then to turn on the ordered transcriptional program. And by that point, it's reactivated, and you're going to go through all of it and make DNA. You're going to make virions. And in fact, it turns out that VP16, here's the last cool part. VP16 helps recruit demethylases to the chromatin to remove all of those methyls. And that's what gives you the final ordered transcriptional program. It's really pretty cool. Now, herpes virologists tell me, yeah, this is great. This is animal models and cell cultures. We don't know if this happens in people. So the caveat is there, but I think it's a great explanation. This is called a phospho switch, a phosphomethyl switch. Putting a phosphate on will reverse the inhibitory effects of methylation of chromatin. The next question is, persistence of herpes simplex in nerve ganglia requires which of the following? Continuous episomal DNA replication, low-level production of virions, silencing of all gene expression of except LAT and microRNA, UV light, stress, or steroids, or all of the above. All right, what's the answer here? The answer is C, silencing of all gene expression except LAT and microRNA. So you don't make virus particles. You could actually get to this answer by eliminating the other things. There's no DNA replication. I told you that like 6 times, right? Because it's a neuron. Persistence doesn't require UV light or stress. That's what reactivates it. So you could arrive at C on your own. But yes, the herpes DNA, it's silenced except for the production of the LAT, which is a small RNA that gets processed to microRNAs that probably regulate host gene expression, and that's part of persistence. A couple more herpes viruses. Epstein-Barr virus is another one that you all have. By the time you get to college, after your first year, you all become seropositive, 95% of US adults. And you also have the EBV genome in your B cells. This is not neurons, this is B lymphocytes. Most of you get infected at an early age. Many of you are asymptomatic, but some of you may develop mononucleosis, okay? That happens in a lot of people who initially get infected, and also some serious diseases, some human cancers, as you can see here. What happens in EBV is, again, you acquire infection early in life or by passage of saliva that has virus in it from your mother or your partner later on. The virus enters the epithelium in your oropharynx. It infects the epithelium, passes through just like herpes, but now instead of hitting a nerve, it infects a B cell. Remember, there are B cells underneath the epithelium that are there in case they need to make antibodies. These B cells get infected. The virus causes them to differentiate and eventually become a memory B cell. The virus makes them become a memory B cell, which lasts a long time. Remember, that's the basis of memory. Eventually, these B cells come to rest in your lymphoid tissue, in peripheral blood, even in the bone marrow, and they are latently infected. Okay, they are making a few viral messages and viral proteins, not unlike herpes viruses. If any of them happen to spontaneously activate, and we don't understand the factors of activation for EBV like we do for herpes, they're cleared by immune responses. But now and then, some of these B cells that reactivate will go up to the skin, go out of the blood vessel, go near the skin, make virus particles, and they will be shed so the virus can infect other people. Right, so a lot less known about activation of this virus, but we do know its reservoir and how we acquire and spread infection. You need B cells for latency. That's where the virus is hanging out. The genome is a self-replicating episome, so if the B cell has to replicate when the virus is activated, the DNA will activate as well. In latency, there are limited repertoire of viral genes that are expressed. As I said, the infected cells, the latently infected cells, again, not producing virus, go to the bone marrow and lymphoid organs. And these cells are not killed unless reactivation occurs. And the virus can escape that initially by modulating MHC, make enough virus to be spread to other individuals. Another herpes virus, Varicella-zoster virus. So when I was a kid, there was no vaccine for this virus, and everybody got chicken pox. All right, so they're the rash of chicken pox, also called varicella, so that's part of the name. You get this infection, again, very similar to the other herpes viruses, by a conjunctiva, upper respiratory tract from your parents or close people. The virus replicates in lymph nodes initially, establishes a viremia. That brings the virus to liver, spleen, and other organs. You get a secondary viremia, which then brings virus to the skin. Virus multiplies in the skin, and that gives you the rash of chickenpox. And this virus, because it's infecting your upper tract, is spread by respiratory secretions to other people. That's why a kid with chickenpox shouldn't be going to school. Of course, nowadays, no one should get chickenpox because we do have a vaccine that works very well. Now, as the virus is infecting the skin, of course, under the skin are nerve endings, just as we talked about for the other viruses. And the virus will enter the nerve endings, go to the neuronal cell body, enter the nucleus, and establish latency in a sensory ganglion. And then the virus remains latent for many years. It will subsequently be reactivated many years later, and that causes a different disease called shingles, or herpes-zoster. There it is there, and these lesions usually occur on your chest, but it can be elsewhere as well, typically in a line that's innervated by a specific spinal root nerve. Okay, so the viruses come out of a specific place and end up being put out the endings of the nerves onto the skin in a pattern according to how the nerves are innervating. And this happens maybe 50 years or more. So before we had vaccine, 99% of adults were infected. 30% of them developed zoster, mostly over 50 years of age. Typically, you get old and you get zoster. I had zoster about 7 years ago. And it's not so bad. I didn't take any antivirals, but in some people it's very painful, and it takes a few months for it to resolve. There's virus in that rash, so you could theoretically spread that to other people. So not only now do we have a chickenpox vaccine for kids, but for people like me who didn't get a chickenpox vaccine as a kid, got chickenpox and is at risk for zoster, there's a zoster vaccine that you can take, and it will prevent zoster. So latency involves episomal DNA replicating, non-replicating in neurons. These cells do not replicate, of course, no need for that. Restriction of viral gene expression, very much like EBV, restricted gene expression, and we don't know really what triggers reactivation to cause the shingles. But this is a virus that's primarily spread at that initial infection, the respiratory spread, and the latency many years later could, in theory, spread, but the main spread is early on. Another herpes virus is cytomegalovirus. Again, most of you, all these viruses that we've talked about, most of you have 99% seroprevalence. This is transmitted by the respiratory routes, virus in saliva, urine, sex. This virus is shed in many different places, can be spread either early, middle, or late in life, replicates in peripheral blood cells and endothelial cells. Here's a graph of seroprevalence of CMV. Different age groups, you can see from 6 to 14 all the way up to 75 and older in a couple of different racial groups. And you can see some have up to 90% and more seropositivity, which increases as you get older. You're more likely to be infected later on in life. The primary infection is usually asymptomatic and replicating in a variety of cells derived from myeloid progenitors, these asterisk cells like monocytes, macrophages, dendritic cells. You shed virus in saliva and urine, and you can do this for months or years, so that's the way virus can infect other people. But that initial infection is eventually resolved, but there are latently infected myeloid cells in your bone marrow, and that's here, common myeloid progenitor. So they become latently infected, they have viral DNA in them, and they will remain there for your lifetime and periodically reactivate, and you will shed more virus in the saliva and the urine. CMV is a problem in organ transplantation, because since everyone is carrying CMV, you get an organ transplant, you're immunosuppressed, this virus will replicate like crazy and could cause very serious disease. So a lot of effort is being directed at trying to figure out how to control infections. We have some antivirals, we need more. It's a particular problem in pregnant women. It crosses the placenta and can cause very severe multi-organ congenital defects and death. So Zika made a big splash when it was found out to cause microcephaly, but we already have a number of viruses that can do this and more. And here's an example of the numbers. If you take 1,000 pregnancies leading to live birth, 1 or 2 of them are going to have babies with permanent problems because of CMV infection, because some of them have CMV already. They're latently infected, and at various times they're going to produce virus. Most of them produce CMV negative babies, but 6 of them will produce a CMV positive baby, and a few of those will have problems. Other women do not have CMV before pregnancy, so this is a smaller cohort. Some of them never get infected and their babies are fine. Some of them do get CMV during the course of their pregnancy, which is very bad, and of them, they give birth to CMV positive babies as well. So there's a lot of concern about this because so many people are infected, and even if you're not infected during pregnancy, there's a good chance that you will become infected, say, from your partner. Final question: what do persistent infections with EBV, VZV, and CMV have in common? A, B cells are essential for latent infection. B, may cause congenital birth defects. C, viral DNA persists as an episome. D, the factors governing reactivation are well known. E, all of the above. The answer is C, viral DNA persists as an episome. And finally, the last 2 viruses are also herpes viruses. These are human herpes viruses 6 and 7. They got tired of giving them names. They just started numbering them. And these viruses cause a childhood rash, pretty mild rash, called exanthem subitum. It's also called 6th disease. There are 6 classic childhood rash diseases, which include measles and a parvovirus rash, some bacterial rashes. Most people get infected with both of these at an early age. They're transmitted through respiratory secretions. Your parents give this to you, very much like many of the other herpes viruses. The virus infects specific cells and becomes latent. And depending on the virus, HHV6 becomes latent in monocytes and macrophages, CD34 plus progenitors, which are stem cells. HHV7 goes into CD4 positive lymphocytes. Now the interesting, and periodically these will be reactivated, and you can spread the virus to others through saliva, for example. If you have a child, you will spread it to your child or to a partner. Now the interesting aspect of HHV6, not 7, but HHV6, is that the viral DNA integrates into the chromosome. And it integrates into the telomere. The telomeres are the ends of chromosomes, right? And they're repeated sequences that get shorter and shorter as you age. We have a telomerase that keeps them long for many years. This virus has a telomerase-like repeat at its ends, so that's the viral genome there for DRL and DRR, direct repeats. And this virus integrates right into the telomere, so in the integrated form, the chromosomally integrated HHV6, you have the end of a chromosome, then you have the viral DNA, and then the telomeres are all the way at the other end. So these chromosomes remain, they maintain their length in the normal ways, but they happen to have an HHV6 integrated. About 1% of this occurs in the germline. The integration occurs in germline DNA. So parents pass the DNA, the viral DNA, onto their kids, who then can make infectious virus without having to be infected. So it's an interesting strategy for latency and transmission. Let me end up with this slide, which was published a number of years ago, showing you the estimated burden of chronic viral infection in humans. So these authors call all of these infections chronic infections. I'd call them persistent. They last your life. So here's the population, how many people have these viruses. And all the way at the right, here's endogenous retroviruses. So that's everybody. So this bar is the entire human population. And then look at these other viruses, anelloviruses. These are little DNA viruses that are in everyone's blood, don't cause any disease. Here are all the herpes viruses we have talked about, all still pretty high up there. Polyomaviruses, adeno-associated virus. Herpes simplex 1 is pretty high, but then we drop for 2. And then, the others are in far fewer people. But look at all these viruses that are in everyone. So 2 points. Persistent viruses are very successful. They can stay in populations for long periods. This is not just in people. Animals have their own collection of persistent viruses. And 2, if someone comes to you, if you're a doc one day and someone comes to you with an affliction, you're probably going to look at their microbiome, their genome, right? Hopefully. You may look at their virome, And if you don't, you'd be wrong, because they're all going to have virus infections which could modulate their cancer or diabetes or whatever disease they have. And that was the point of this article, to bring up the fact that we have to consider everything, all the infections we have together, because they could modulate other illnesses as well.